Epigenetics is a study of inheritable gene regulation. This is essentially the study of non-mutational modifications to DNA sequences. DNA sequences can be modified non-mutationally by several means, but the most common would be DNA methylation. Methylation is the addition of a methyl group to a molecule, while another means of modification is hydroxymethylation, which is the subsequent addition of a hydroxyl group to the to a methylated DNA sequence. Methylation and hydroxymethylation are illustrated in this figure, where we have cytosine, methylcytosine, which is methylated cytosine, hydroxymethylcytosine, which is hydroxymethylated cytosine, and for comparison, thymines, you can see that the essential structure of the bases are not changed in the non-mutational modification. Methylation in the epigenome, as well as hydroxymethylation, can lead to gene silencing, but they can also have an impact on cancer. In my X chromosome inactivation study, or my XCI study, embryonic stem cells from mice were placed into LIF, and 20 nanograms of complementary DNA, or cDNA, were created using this method illustrated in this figure. The DNA was analyzed for allylic ratio consistency. Consistent DNA was uploaded onto the GSNAP algorithm and mapped. Then in my structure modification study, 12 base pair sequences were modified in the 3rd position, the 9th position, or both positions, as well as and the wild type version of this sequence was also analyzed. This wild type sequence was taken from the from the protein database, the protein data bank, or PDB. The 20 base pair sequence was created on the X3 DNA program, and we looked at them. The researchers looked at the modified, the wild type sequence, and the sequence that was modified at the 10th base. All modifications in this study were to the cytosine base.
My 2 studies were on the effects of methylation of the epigenome and in cancer. The first study was specifically on colon cancer. So they used RNA from the cancer cell line HCT lysosine and abnormal adjacent cells, as well as 30 other cancer cell lines with 208 primary tumors and 22 corresponding normal tissues. The RNA was microarrayed to normalize miRNA expression, and the CpG sites with the miRNA was determined through PCR on the bisulfite modified DNA. Western blot analysis was performed for cyclin dependent kinase 6, or CDK6, and retinoblastoma, or RB expression. The other study was on breast cancer. The RNA was extracted from 20 pairs of tumors and adjacent normal cells, and PCR was performed on the resulting cDNA. The bisulfite sequencing method was used to determine CpG sites of Bessel-1 methylation, and BRCA1 expression was addressed using anti-BRCA1 monoclonal antibody samples, and positive staining was determined at 10% staining.
In my XCI study, the k-means clustering found forest clusters of genes. There were genes silenced at an early stage, silenced at an intermediate stage, and silenced at the late stage, as well as unsilenced genes. Unsilenced genes were correlated with specific topologically associating domains, or TADs. In my study on DNA modification, methylation was found to affect twist angles in 3 DNA, or DNA methylated at the 3rd position, roll angles in 9 DNA, or DNA methylated at the 9th position, and in the 3rd, and the 3-9 DNA methylated at the 3rd and 9th position, was mostly just the sum of the effects of the methylation at the 3 DNA and 9 DNA added together. but there was some differences in the tilt-roll angles for the 3-9 DNA as compared to the sun, and you can see these differences in this figure. Okay. And
what? Okay. In the colon cancer study, the CPG island for MIR124A was methylated in HCT 116 cells and was found unmethylated in normal cells. The MI124A hypermethylation was found in colon cancer as well as breast and lung carcinomas, leukemia, and lymphomas, as shown in this figure down here, showing the differences of methylated and unmethylated in these different kinds of cancer. The CDK6 and RB phosphorylation was associated with the MIR124A hypermethylation, as shown in this figure here. As you can see in the methylated section of these cells, there is strong expression and phosphorylation. In the breast cancer study, the downregulation of Bessel-1 was found in 14 out of 20 tumors. 6 out of 20 tumors shows significant decrease of Bessel-1 mRNA expression. This is shown in figure 6. And shown the brown color here is showing positive Bessel-1 staining. A is showing normal tissues, B is showing a breast tumor. And as you can see with the decrease in brown, there is less expression. The BRCA1 positive staining was found in 6 out of 20 tumors, and the Bessel-1 mRNA expression was significantly higher in the BRCA1 positive tumors.
The XCI study showed that specific TADs were correlated with XCI propagation, while XCI propagation avoided other specific TADs. My study on DNA structures and DNA structural modification showed that modification affected twist, roll, and roll/tilt angles in different modifications of DNA. This can affect the genomic stability, which could lead to silencing or to cancer.
In the colon cancer study, the methylation of MIR124A leads to strong expression of CDK6 and RB phosphorylation, which may lead to cancer. The hypermethylation of the MIR124A was found in other types of cancer. And the methylation of mRNA in cancer is an emerging field and could impact patients that are taking demethylating drugs for treatment. And in the breast cancer study, Bessel-1 was found to have decreased regulation in breast tumors, and the Bessel-1 expression in the BRCA1 positive tumors may suggest that the Bessel-1 expression may be linked to breast cancer's growth.
Overall, we found that TADs lead to silencing of genes. Methylation through modification can lead to changes in expression, and microRNA and gene methylation is correlated with cancer.
Here are our references, and thank you.